Hello and welcome to Learning Made Easy. Today we're going to be discussing the complement system and its three pathways, the classical, alternate, and mannose-binding lectin, MBL, pathways. The complement system is a crucial component of our immune system that helps to defend our bodies against pathogens. The classical complement pathway is one of three pathways that activate the complement system, which is a part of the immune system. 1. The pathway is activated by antigen-antibody complexes with the antibody isotypes IgG and IgM, as well as by apoptotic cells, necrotic cells, and acute phase proteins. 2. The pathway starts with the binding of antigen-antibody complexes to the C1q protein. The globular regions of C1q recognize and bind to the Fc region of antibody isotypes IgG or IgM. 3. The binding of C1q leads to conformational changes and the activation of the serine protease C1r, which then cleaves and activates C1s. Activated C1s cleaves C4 into C4a and C4b. 4. The newly formed C4b is deactivated by the cleavage of its thioester bond by water. C4b is restricted to only bind to pathogen surfaces and is rapidly deactivated. 5. Surface-bound C4b acts as a receptor for the binding of C2, which results in the formation of C3 convertase, composed of C2b and C4b. C4a and C2b act as potent cytokines, with C4a causing degranulation of mast cells and basophils and C2b acting to increase vascular permeability. 6. C3 convertase cleaves many molecules of C3 into Ca and Seb, with Seb acting as an opsonine and recruiting phagocytes to recognize and engulf pathogens. 7. Seb binds to the C3 convertase to form C5 convertase, which cleaves C5 into C5a and C5b. C5a is an anaphylatoxin that attracts leukocytes. 8. C5b initiates the formation of the membrane attack complex, MAC, by binding to C6, C7, C8 and multiple molecules of C9, which ultimately creates a hole in the cell membrane of the pathogen, leading to the loss of essential molecules and eventually leading to the death of the pathogen. The mannose-binding lectin pathway or lectin pathway is a type of cascade reaction in the complement system, similar to the classical complement pathway. 1. MBL pathway does not recognize an antibody bound to its target. 2. The lectin pathway starts with mannose-binding lectin, MBL, or ficillin binding to certain sugars. 3. MBL binds to mannose, glucose, or other sugars with 3 and 4 O groups placed in the equatorial plane, in terminal positions on carbohydrate or glycoprotein components of microorganisms, including bacteria such as Salmonella, Listeria, and Neisseria strains, and some viruses such as HIV-1 and respiratory syncytial virus, RSV. 4. MBL is a protein produced by the liver that can initiate the complement cascade by binding to pathogen surfaces. 5. MBL forms oligomers of subunits, which are trimers, and forms a complex with MASP1, mannose-binding lectin-associated serine protease, MASP2, and MASP3, which are protease cymogens. 6. When the carbohydrate-recognizing heads of MBL bind to specifically arranged mannose residues on the surface of a pathogen, MASP1 and MASP2 are activated to cleave complement components C4 and C2 into C4a, C4b, C2a, and C2b. 7C4b tends to bind to bacterial cell membranes, and if not inactivated, will combine with C2A to form the classical C3 convertase, C4bC2A, or alternative C3 convertase, CEB, on surface of pathogen. 8. After the formation of the C3 convertase, the next step in the lectin pathway is the cleavage of C3, a central component of the complement system, into CEA and CEB. 9. CEB also plays a key role in the formation of the C5 convertase, which cleaves C5 into C5A and C5B. 10. C5b initiates the formation of the membrane attack complex, MAC, by binding to C6, C7, C8 and multiple molecules of C9, which ultimately creates a hole in the cell membrane of the pathogen, leading to the loss of essential molecules and eventually leading to the death of the pathogen. The alternate pathway, also known as the Perperdin pathway, begins with the spontaneous hydrolysis of C3, which results in the formation of CEB. 1. CEB then binds to the surface glycoprotein of pathogens, which is not masked with sialic acid as it is on host cells. 2. The binding of CEB to a microbe causes a change in shape, which allows the binding of plasma protein factor B. This, in turn, allows factor D to cleave factor B into BA and BB. 3. BB remains bound to C3H2O to form C3H2OBB, also known as a fluid phase C3 convertase. 4. The complex is believed to be unstable until it binds perperdin, a serum protein. 5. This addition forms the complex CEB, a stable compound which can bind an additional CEB to form the alternative pathway C5 convertase. 6. The C5 convertase of the alternative pathway consists of CEB2BBP, which can cleave C5 into C5A and C5B. 7. C5b binds sequentially to C6, C7, C8 and then to multiple molecules of C9 to form membrane attack complex. In summary, the complement system is an important part of the immune system that helps to protect our bodies against pathogens. It has 3 pathways: the classical, alternate and mannose-binding lectin, MBL, pathways. The classical pathway is activated by the binding of C1q complement protein to the antibodies bound to antigens on the surface of pathogens. The alternate pathway is activated by the spontaneous hydrolysis of C3 complement protein. The mannose-binding lectin, MBL, pathway is activated when mannose-binding lectin, MBL, binds to the carbohydrates present on the surface of pathogens. Each pathway plays an important role in the body's immune response and works together to defend the body against pathogens. We hope you found this video informative, and thank you for watching Learning Made Easy. Don't forget to like this video and subscribe to our channel for more educational videos on a variety of topics.